As scientists worldwide race to develop an effective coronavirus vaccine, research has revealed that the virus invades cells through spike proteins, which have specific characteristics that are important to understand. Saville reports from Imperial College London that vital research looking at these spike proteins may unlock the door to improving testing and finding a successful vaccine.
At Imperial College London, one of the largest and most research-active life sciences departments in the UK, multiple disciplines are addressing one of the most pressing real-world problems, the COVID-19 pandemic. Studying each level, from molecules to ecosystems, the work here could lead to better diagnostics and a vaccine. Structural biologist Professor Stuart Haslam is investigating the sugar coatings on spike proteins on the surface of the SARS-CoV-2 virus, which causes the disease COVID-19. He says the sugar coating, or glycoprotein, is a recognition molecule that binds to receptors on the human cell.
Our research is showing that these sugar molecules on the virus and on the host molecule come into very close proximity. So it might be that there are interactions going on between the sugar molecules and that variations in the sugar molecules on both the virus and the host can change the strength of the interaction and potentially the infectivity. One of the functions of these sugars from the virus perspective is it's actually acting as a protective shield on the important functional spike protein. So by having an outer layer of sugar around the functional protein, it's masking itself from our immune system, allowing it to infect our host cells.
Coronavirus-derived samples are loaded into an analytical tool called a mass spectrometer, which characterises the structure of their molecules, helping understanding of their function. Dr. Anya Kruger, who worked on a pig virus vaccine in Thailand in a UK-sponsored partnership, says the role of glycans, carbohydrate-based oligomers, could be crucial.
We are really learning something about the diversity of glycans on the spike glycoprotein, for example. And the diversity can make a difference in terms of recognition by the immune system. For example, monoclonal antibodies may bind differently to the virus or to the spike protein due to the fact that glycans could cover also the underlying amino acids. We would really try to see if we can use this information to develop a vaccine.
From lab work to field work, here at the Department of Life Sciences campus in Berkshire, these huge mesocosms, or outdoor experimental systems, are testing just how long synthetic copies of the SARS-CoV-2 RNA can survive in various conditions. Will it remain in the environment around us? Each tank has a different temperature and level of pollution. The teams track how quickly the RNA of the virus degrades to model its persistence in natural environments. They're also testing samples of sewage to assess the extent of the virus in local populations and checking animal droppings.
One of the things that we're a little bit worried about is that if SARS-CoV-2 ends up in wildlife populations, then it might actually be able to transfer back to humans at a later stage, especially through organisms like bats, who actually live in roofs of houses, and so their locality to humans is actually quite close.
At the Silwood Park campus, extensive microbial research facilities bring together a range of expertise.
Here in-house, we're less of a department really, and more of a multidisciplinary team. We have experts in genetic and molecular techniques, ecosystem level manipulations, experimental designs, big data analysis and theory. Evolutionary biology gives us a context to how every species interacts with every other one and its environment, including viruses, so that we can be prepared and we can mitigate and proactively stop things like this happening again in the future.
That's the wider challenge underlying the bioscience research at Imperial College London, working to protect humankind from existential threats in the world around us.